So good morning, all of you. We had started with some points of genetics. We did this basic ideas. We're going to continue from there. If you have any questions from what we did in the last class, you can ask here. We will start with one question from the topic covered yesterday. This is a question from the endocrine system part, which we did yesterday. So, assertion reason type question. See if you can connect with this. The question is, assertion, the person with diabetes insipidus feels thirsty. This is the first statement. The second one is, a person with diabetes insipidus suffers from excess secretions of vasopressin. So, the disease they have mentioned is diabetes insipidus, and the reason for that they have given is excess secretion of vasopressin. Right? And the answers which I am getting are B and A. C also. Okay, A, B, C, all answers you have given me. Let's see. Now, the person with diabetes insipidus feels thirsty. Diabetes insipidus is a disease of the deficiency of ADH. ADH or vasopressin, its deficiency is what causes diabetes insipidus and not its axis. So reason in this way is false. The reason is not correct. Reason is false. And out of these four options, there is only one option in which the reason is false. That is C. So in diabetes insipidus, what happens? ADA's job is to reabsorb water and nutrients from your kidneys. So if they do not do it, their job, if it doesn't reabsorb water into back from the kidneys back into the system, then that means there is going to be more loss of water. And with more loss of water, what you are going to have is you will feel dehydrated and you will be thirsty all the time. So the assertion is absolutely correct. The reason is wrong because it's not excess of vasopressin, it is its deficiency. Okay. So answer is C. After this, let's go to our genetics part. We are going to talk about the phenomena of determination of sex. How that idea of being a male or female is established by the help of hormones. So, sorry, with the help of chromosomes, I'm sorry. So, we're doing the question on hormones. So, when we talk about our chromosomes, we did this yesterday that there are total 23 pairs of chromosomes. Okay, we have a total of 23 pairs of chromosomes. Out of these 23, 22 pairs are called autosomes, and there is one pair which is called allosome or the sex chromosome. Please write this down. 22 pairs of chromosomes. They are called autosomes, and there is one pair which is called the sex chromosome, and this is going to be the one which will determine the sex of a person. In this pair, we have X and X. There are two chromosomes basically, X and Y. If it is X and X, we have a female. If it is X and Y in the combination, or everything is in pairs, right? Then that would be a male. And how they are coming in, how these chromosomes are reflecting, that is what will determine the sex of a person. Let's take a look at that cross which is there. And by now, because we have already done some crosses in the last class, I think this should make sense to you. So we have the father with X and Y. So we can do another step of gamete formation here. X and Y separate. Then for mother, X and X. Checking for different combinations, you have a daughter, a son, a daughter and a son. So if we look at the ratio here, it will be 1:1 or 50% for both boys and girls. This means there is an equal probability. When a child is born in nature, there is an equal possibility of the child being a male or a female. The gametes or the eggs which are coming from the mother, they are all the same. They will all have X chromosome. There is not much choice here. So ultimately, what kind of a sperm will come and fertilize this egg, that will determine the outcome. Is the sperm having an X chromosome or is it having a Y chromosome? That is what is going to ultimately lead to the sex of the child. So it's an equal probability, right? And you know, if there's this concept of preference for male child in our society. So whenever that problem is and it's not working out in the way some people expect it to be, so usually they blame the mother that you cannot bear us a son. right? So maybe the next time if you ever come across a conversation like that, you can explain this inheritance pattern to them, that it's actually the sperm which will decide. वैसे तो this is nature, nobody can decide. It's something which is happening from with the help of a force beyond our beyond us. Okay, the father or the mother, nobody can control it, but nevertheless, if still technically you have to put it somewhere, it has to be put on the sperm. All right. I hope you have drawn this and this is understood. We are going to talk about genetic diseases after this. Some kinds of disorders which are associated with the different patterns of inheritance. The concepts of Mendel here also are going to be very useful because it's going to be based on that. The first type of genetic abnormality or genetic problems, that is what we are going to discuss is going to be the defects of chromosome number. Now this chromosome number, this we said is fixed for a particular species, right? Human beings are supposed to have 46 chromosomes. If there is any alteration, if there is any gap, if there is any problem with this 46, sorry, not being 46, then that would reflect in some kind of an abnormality. Which chromosome is extra, that will have, or which chromosome is less, that will have, or that will manifest in the form of different diseases. The first example which we are going to take is Klinefelter's syndrome. Klinefelter syndrome. In this, what we have is, we have X, X and Y. So we have an extra X chromosome in this male. This is going to be a male and with one extra X chromosome. So the total number of chromosomes here are 47. Instead of 46, which is normally supposed to be, the number of chromosomes here are 47. Okay. And this is a male, and this male person will develop some feminine characters also, like enlargement of breasts, sparse body hair, or a high-pitched voice. The basic secondary female characteristics this person is also going to have because of this presence of extra X chromosome. So you can just write this in one line where you've written Klinefelter syndrome. Just write down with that. This is a male. This is a male with development of feminine characteristics. This is a male with development of feminine characteristic. Just like this was axis, we can also have a situation of deficiency. One chromosome less. That means 46 chromosome, and the condition is called Turner syndrome. In this is a female, but instead of 2 X chromosomes, there is only 1 X chromosome. This this is actually 0, indicating that this space is empty. So instead of 2 sex chromosomes in a person, Turner syndrome female only has 1. and she is reproductively sterile. He cannot reproduce. So you can just write this with Turner syndrome. One missing chromosome, one missing X chromosome, and a sterile female. So all those people which we understand as eunuchs, which are there, so they can be amongst these people. Okay, they can be the ones who have chromosomal abnormalities, but that is not the only condition which can lead to a situation like that. We can also have deficiencies of hormones, the sex hormones which are produced in the body. There can be abnormality in their levels also, because of which people can become those eunuchs, right? Other than that, there are other fluid sexual identities these days, fluid gender identities. People are non-conforming, or the concept of transgenders, or people who want to change their gender. So a lot of those conditions, or a lot of those people might not have any biological condition. Biologically, they might just be a normal male or a normal female with appropriate levels of hormones, appropriate chromosomes, but it can be an issue of a psychological aspect. That psychologically, like you might have heard some people say that, I was born as a male, but I always felt that I was a female inside. Without any chromosomal or hormonal abnormality, it could be a thing of the psychological aspect also, right? There is a difference between a sex and the gender. Sex is biologically what the chromosomes tell us. So is it XX, is it XY? What is the condition? That is the word we use for that is sex. Gender is more of a social construct. Okay, socially, there are certain attributes which are associated with female. There are certain other attributes which are associated with males, right? And this, that the society has defined the idea of being a man or being a woman. So that gender identity, that a person can choose, again depending on psychologically or mentally how he wants to associate or he doesn't want to associate. There are a lot of people who do not want to associate with any of these ideas. Gender non-conforming identities are also there. They don't even use the prepositions he, she, that also they do not use. So it or them, something like that, they'll use. Maybe you've come across these ideas, and that distinction between gender and sex should be there. In terms of sexual abnormalities, we can have abnormality on the chromosome front or on the hormonal front. Okay. Then one more example we'll do in this genetic, in the chromosome number abnormality, and that we will do from an autosome. That means other than sex chromosome, another chromosome, and the disease is Down syndrome. This is because of the extra chromosome number 21. It is also called trisomy 21. Okay. See about LGBTQA, right? I have explained one aspect of people having non-conforming gender identities or psychologically not being without any abnormality associating with a different gender. As far as the homosexuality concept is concerned, that doesn't comply with any of these. Okay, that is more of a sexual preference which somebody might have. It has nothing to do with how their chromosomes are acting. So people can be following homosexually, that's just a sexual orientation or the way they have their sexual preference. Okay, so people can be lesbians or gays or bisexuals, or there are many, many more such terms also which are coming in, but that is just to do with the preference which they have in terms of how they want to sexually interact with another person, but it has nothing to do basically with their biological systems. Their biological systems and everything about them could be uh, perfectly in place. It is not an abnormality in any form. Okay. Let me show you an image which depicts Down syndrome, so that we see the picture of the chromosomes. So this is the basic depiction of the chromosome pairs. We spoke about homologous chromosomes. So these are the how the pairing takes place. We have 23 pairs, and in that when we talked about homologous chromosomes, we said they are of similar size, structure, genes, patterns. So you can see they have variable shapes, and those with similar shapes and sizes, they are the ones who have come together. Okay, so we also spoke about the fact that we are going to be numbering our chromosomes 1, 2, 3 like that. So 1 to 22, and the last one is the sex chromosome, so XX or XY. Okay, now in this 21 pair, we have one extra chromosome. Presence of one extra chromosome in chromosome number 21, that is what is happening in Down syndrome or trisomy of 21. Okay, see, if instead of 21 chromosomes, some other chromosome was extra, we would have a different disease. Depending on which chromosome is extra, we have different disorders. There are many other diseases also. We've just taken this one example. Okay, so you can write this down, just one line about Down syndrome. Down syndrome. Down syndrome is caused by presence of an extra chromosome number 21. Presence of an extra chromosome number 21 called trisomy 21. Called trisomy of 21. The child born with Down syndrome, has a small rounded head, small round head, partially open mouth, and mental retardation. Small round head, partially open mouth, and mental retardation. So the children with Down syndrome, they are actually very easily recognized. You can very distinctly see how they look. Okay. YO cannot be possible because Y is actually a very small chromosome, and without X alone it cannot survive. You can see here the size between X and Y chromosome is very small. So Y alone, we do not have a surviving organism in that case. Now, after this, the next thing we are going to talk about is gene defects. After defects of chromosome number, defects in certain genes we are going to discuss. The first disease which we have to talk about is an autosomal disease. The example which we are considering here is sickle cell anemia. So this is a disease in which what happens is the shape of red blood cells changes. So red blood cells, which we said are supposed to be biconcave, they will become something like this, sickle-like. Sickle is a farm instrument. So the shape of red blood cells becomes sickle-like because of the defect on one of the genes on the autosomal chromosome. We are going to study the inheritance pattern of sickle cell anemia. It is based exactly on the principles of Mendel's inheritance. The same way this is going to happen. So, let us call the gene which we are considering here to be HB. Why HB? Because we are associating it with red blood cells. So I've used HB. HB का कोई significance नहीं है. You can call it X, Y, Z, whatever you want to call it, you can call. So in every organism, there is a pair. For every character, we have a pair which is supposed to be there. So this is a normal individual. A normal person with two normal genes. This is the gene which we are considering, which if defective is going to be responsible for causing sickle cell anemia. Okay. Now, the defective gene we will call HBS. So, a person with two defective genes will have sickle cell anemia. Two defective genes, they will have sickle cell anemia. Okay. Now, there is also a possibility of a heterozygous state where one gene is normal and one gene is abnormal. Okay, one gene is normal, one is defective. That is also a possibility. This disease and most of the genetic diseases, they are recessive. What do we mean by being recessive is that the abnormal gene, the defective gene is going to be recessive to the normal gene. Remember T and T? We did this yesterday. So when T and T were together, we said capital T is dominant, so the plant is going to be tall despite the presence of the dwarf gene. So here also same thing. Between normal and abnormal gene, the normal gene is going to be dominant and the recessive will not be able to play any part. So this person, even with a defective gene, is going to be normal because it has a normal gene which dominates and overpowers the effect of the defective gene. Okay. However, this person can still carry the disease to the next generation. See, somebody who has a disease, you obviously know that, okay, this is going to be transmitted to the next generation. Because the person is suffering from the disease, the person has the disease. But the person who with one defective gene is not suffering from the disease, but it can still pass on the disease. Think of this situation where there are two people with one defective gene for sickle cell anemia. They are not suffering from the disease, but they can have children who can be having sickle cell anemia. They can have children with sickle cell anemia because both of them will pass on their defective gene to the child. Right? So, in this case, we call this person with one defective gene as the carrier. He is normal in his own self, but can still be the carrier of the disease. Okay, simple aspects of Mendel's inheritance, which we already have studied. Normal with both normal genes, with both the genes defective. Sickle cell anemia, one gene normal, one defective. This is an autosomal recessive disease. So, in this way, we have the person being normal but a carrier of the disease. Normal because the normal gene dominates over the effect of the defective gene. Okay. Let's now talk about gene defects on the sex chromosome. This was on the autosome. Now, when it will happen on the sex chromosomes, we will also be talking about how the sex of the person is involved in that. So, we can call this sex-linked disorders. And the examples, the most common examples are color blindness, that is the inability of a person to distinguish between the primary colors, and hemophilia. What is hemophilia? Hemophilia is basically a disease in which clotting of blood is affected. The blood clot which is supposed to be forming, that does not happen properly. And why is that? It is because of a gene which is defective. So these people who have hemophilia, they are very susceptible to any kind of blood loss. So even with the most minor injuries like, a paper cut or something, their blood will not stop. They will continue having a blood clot. Okay, so the clotting of blood continues in these. The clotting of blood is delayed. That's what is hemophilia. And this was there in Queen Victoria, and the gene, defective gene, is still running in her family, in Queen Victoria's family. So hemophilia is also called royal hemophilia. Okay. Let's now see about the gene pattern. So you have XX and XY, normal male and female. This we already know. XH, XH, hemophilic female, two genes defected. In male, this is a disease which is associated with the X chromosome. This is a disease of the defect on the X chromosome. So, along with being sex-linked, we can also specifically call it X-linked disease. See, X chromosome is the bigger chromosome, is the larger chromosome. It is the one which has more number of genes. So most of the diseases, the sex-linked genetic diseases, they will be on the X chromosome. There are some diseases, however, on the Y chromosome also, but more often than not, most of the sex-linked diseases are on your X chromosome only, right? And when it comes to males, they have only one X chromosome. So when that one and only X chromosome is defective, they get the disease. Okay. the gene on X chromosome, there is only one X chromosome. Remember this. Y, I have told you already that Y is a very small chromosome with very few number of genes. So Y cannot counter the effect of X. Right? So with one defect, please remember this. This is where your questions will go wrong. So with one defective X chromosome, the males suffer from X-linked diseases because they do not have another chromosome to counter its effect. Okay, so these are the two combinations of a diseased person, male and female, and sorry, the heterozygous state makes sense for the female because she has two X chromosomes, so one normal X chromosome, one defective chromosome. This is also a recessive disease. Most of your genetic diseases are recessive. There can be dominant diseases also, but that's very, very rare. Okay. So, sex-linked recessive disease. Recessive means the normal X chromosome will dominate over the defective X chromosome. So, here we can say this is the person, the female is normal, but at the same time, she will be the carrier of the disease. Normal but carrier. Please make sure you've copied all this. Nitya has asked, the person suffering from autosomal can have blood transfusion with a normal person. Yes, it has nothing to do with blood transfusion. See, unless and until the disease condition itself, like sickle cell anemia mein, what we had abnormal red blood cells. So obviously a person like that will not be able to give blood because the RBCs are defective and you don't want to send defective RBCs to somebody else's body. But if this is any other kind of disease which is not associated with blood, then very much you can give blood transfusion if the blood levels are normal. Okay, depending on where the abnormality is lying, based on that, you will do the transaction. Can humans have genotype BB or BO? Okay, Mukul, if you are asking this about blood groups, I'm not sure in which context you are asking. So blood group में तो BB और BO होता ही है. If you're talking about something else, please let me know that. But when it comes to your blood groups, if that is your zone, then in blood groups we do have BB and BO, which are going to be specifically the blood group B. Right? I think we wrote this yesterday. So if there is some other aspect which you are confused about, please ask again. We have everything. We have A, we have B, then we have AB, and we also have O. Please ask your doubts in the doubts section so that they can properly reach us and we can address them. We will solve one question based on this. Just to let you see if you have understood the concept. Please answer this question. If you're asking about both BB and BO, if that was the question and I didn't get the sense of it, then that's not possible. No person can have two genotypes. Okay, the genotype for one character would be one out of two, either BB or BO, not both, if that is the sense of the question. It becomes a little difficult to actually understand the question sometimes. It's either of the two. Okay, so the answers which you are giving are D in most of the cases. Okay, let's see. Please try and solve these questions only with the help of a cross. You will always get the correct answer if you are making the proper cross to talk about their inheritance. In the context of genetic disorders, consider the following. A woman suffers from color blindness while her husband does not. So we know the two parents. Let's take a look at their children. So you have a woman who is suffering from color blindness. So this is going to be her genetic composition. Then the father does not have color blindness. So this will be XC for the mother and X and Y for the father. Now, gametes formation or segregation of these traits and then checking for different possibilities. X and XC, then XCY and XCY. So we have the daughters with one normal chromosome, one defective. They would be normal because this is a recessive disorder, and in this case males just with one defective gene, they will have the disease because there is only one X chromosome and that X chromosome itself is defective, so they get the disease. So this tells us that your sons will be colorblind while the daughter does not suffer from it, which is your option D. Option D, as told by most of you, it is correct. Okay. In terms of treatment aspect of these disorders, so the abnormality in chromosome, as of now, we have no way of correcting that, right? But the gene defects, we have developed some technologies in which we can do an alteration in the genes and rectify them, right? But chromosomal number abnormality. See, we have a possibility of giving new genes, creating, editing, forming genes, but we do not have the capacity of forming a whole new functional chromosome, right? We can make changes at the level of the genes, but not at the chromosomal level. We cannot like create a whole new chromosome. That's not possible. We can identify these diseases. Nowadays, very commonly in India also, genetic tests are done while in the period of pregnancy to check if there are any abnormalities, but there is no solution. If you find out that there's a child who would be having Down syndrome, so you do not have any solution. The only option with the parents is that they can terminate their pregnancy. But as far as the correction is concerned, for genes, we can do, if there's just one individual gene which is involved, but not for the chromosome number. Okay. One more query I have. But there are Shashank has asked, there are homologous N, so why must counter X? No, actually this concept of, I understand the logic with which you are coming, but X and Y are not equal chromosomes. There, I understand your idea of the homologous chromosomes, but that rule does not apply with X and Y. Okay, X and Y are not similar chromosomes. That is the speciality, and you can say that is the exception to this concept of homologous chromosomes. So between X and Y, you can see this also in shape and size, because there are sex chromosomes, and that distinguishing aspect of creating the sex for different person, the idea of having a male and a female, because of that these chromosomes are different. You can visibly see them to be different. X is actually a very, very large chromosome as compared to the Y chromosome. Okay, that is why that effect of countering is not there in nature. Because it is smaller, it has lesser number of genes. And even if you forget about this counter effect or any aspect, there is a disease which is for X chromosome. So X ke liye you need another X to counter it, because you will have to be needing the similar genes, right? Then only the job can be done. Okay. Now, after this, we are going to talk about different aspects in which we can manipulate this whole idea, or the concept of biotechnology, basically you can say this. So, we have our genetic material, we have discussed the concept of DNA. Now we are going to see how we can make changes, right? How we can make changes. And if we talk about the word biotechnology, this basically means that we are doing some form of genetic engineering. That means we are doing some alterations in our genetic material. And whenever there is this word technology involved anywhere, it means that this is an application for welfare of humans. So some positive use you need to put that into, that's when the term technology comes into play. Okay, so biotechnology is basically genetic engineering which you will do, or some changes in genes which you will do for the welfare of humans. Now, the thing for you to consider here is that there is a universal nature of DNA. DNA everywhere in all organisms, right from bacteria to fungi to plants to animals. Everywhere your DNA chemically is the same. If you remember, we discussed that DNA is made up of nucleotides, which are made up of nitrogenous bases, phosphate group, and ribose sugar. So that is standard everywhere. You get it from any source, that is going to be the composition. But because of this similar or universal nature of complication, it is possible for us to add and subtract this genetic material. We can add, subtract, join the DNA from different sources because chemically the nature of DNA is the same. Okay. We'll start with talking about different technologies. There are 3-4 technologies which we have to talk about, right? This is an aspect, as a topic, this is asked in the mains exam also. But the question there are not going to be the science kind of questions. The main question, the main theme which they have explored, which you can develop in terms of points, is this welfare aspect. Okay, so they will ask how this is going to help. Okay, like last year they asked a question on how it will add to farmers' income. This year they have asked how this is going to help the lower socio-economic status people. This is the question. I hope you've made the effort of going through the mains question papers, the exam which happened just 2-3 days ago, right? If not, that's something you should ideally be doing. I know you've not covered all the syllabus, but just to get familiar with what's happening. Okay. So, this aspect, how you are going to prepare and from where you are going to prepare is, you are going to have a lot of this covered in the current affairs. Okay, you will have a lot of this covered in the current affairs. One source which you can refer right now, which I want to tell you because it is relevant and it is updated as of now. Further, you have to obviously keep updating yourself, but there is a document called Mains 365. Vision IS releases this document. Okay, it was released recently for this mains, the people who were appearing, right? And it covers September to September. And in this sense, because it is written with the orientation of the mains, it is going to have a very good theoretical aspect, like explaining what is biotechnology, what are its types, what are other theoretical aspects. So in this, the science and tech section mein, that is one document which you can refer. Basic theory remains the same. Of course, with current affairs, as you read them each and every month, you have to keep adding to it. You will read articles and editorials also talking about the benefits of biotechnology. Then you sometimes have, you might have heard about these, and some of you might also be reading these. Okay. See, Mains 365 will come again when you write mains, but just now to start with the base, this is good. The disease part also they have done well, the biotechnology part also they have compiled well. So along with these classes, this can be a very good concised compiled notes for you. Okay. Then Yojana and Kurukshetra are two government publications which are released on different topics. So many a times what happens is that I'm not suggesting that you read all Yojanas and Kurukshetra, whatever is available, but pick out the topics which are linked to your syllabus. That is why knowing the exact syllabus is very important. You need to know exactly which paper, GS1, 2, 3, 4, where, what topic is there. So that when you read a heading, that this is going to be on one topic, right? So if that topic is linked to your preparation, to your subject, then from that magazine, you can read 1 or 2 articles. There'll be like 10-15 articles, but obviously you don't need to read all of them. But read 1 or 2 articles to get sufficient content. You can make notes out of that or add it to your already existing notes. Like maybe there is a topic biotechnology in health or biotechnology for aquatic organisms or food, biotechnology in food processing, or genetic engineering for the aid of the farmers. There are topics like these which keep coming in these magazines, and they can also become very good sources for you to get points. Basic aspects, आपको क्या चाहिए? You have to talk about how it is helpful, what are the challenges. I'll talk about a little bit about different aspects on these ideas also, but just an overview I'm taking, right? So, question का advantage एक और है मेंस में. With biotechnology, this is just one small part of the elaborate GS3 paper, but one question is asked. We've seen this trend that they ask one question, or that is a very broad based question, not a very specific question. Like they will not ask you to explain a particular technology in detail in different aspects. So science wala part they ask less. Mostly they'll ask the welfare part. And in that, it is your discretion. How specific do you want to get? Like if they just ask you, how will biotechnology or biotechnology also they have not asked. They've just asked, how can technology be used to increase farm incomes? And this is exactly a mains ka topic. Technology to the aid of farmers, something like that. Okay. Now in this, you can add many things, everything from marketing to government subsidies to use of fertilizers, and along with that, aspects of biotechnology and use of genetically modified crops that also you can add. It's on you how much specific and how much content you have. So reading and building that content through your current affairs is very, very important. Okay, so keep doing that. The science part of it, the technologies, how they are working, what DNA manipulations are taking place, that we will do in the class. This part you will manage, you will prepare on your own, and you will keep preparing, because this is something which you will keep getting ideas as you need more and more newspapers. Okay, but just as a reference, this I have told. Okay. Now coming to the first idea of the technology which we are going to do, it is going to be recombinant DNA technology. So I told you that there is a, okay, I have a question. Can Klinefelter's and Turner syndrome be affected by color blindness? Okay, this is actually, Ajit, a very interesting way of thinking, but I don't think I have read about any case study or anything in specific. But technically, if I think about it, I think yes, why not? Very much a gene defect in your X chromosome, that can very much happen, and that can very easily be happening. Okay, I don't think there's an impediment to that. Okay, Hari has asked vitamin A deficiency, retinitis pigmentosa. Okay, is it genetic? In the gene point of view, is this disease carried by genes? See, if we have already discussed something to be a vitamin A deficiency, why would you want to actually confuse it here with a gene defect? There is definitely a possibility of a gene defect being associated with some form of deficiency, which leads to, your body becoming less receptive to some vitamins. That can definitely happen, right? But I don't think that is the zone in which you should be getting into. It's a good thing you know about retinitis pigmentosa, but let's not complicate things where we don't need to. Okay. Vitamin deficiency disease को vitamin deficiencies के लिए पढ़ा है. That's relevant. You know it is happening because of vitamin A deficiency. So that is correct. But there we are mainly talking about the nutritional aspect. Genetic भी हो सकता है. I'm not ruling that out, right? But That should not be something bothering you. Okay, here. So, we are trying to create a combination of DNA here. Okay, we are trying to take DNA from two sources and create a new DNA. Okay, so there is one DNA fragment. This is the main fragment which we are interested in. And we are going to take this, put this in another piece, join them and create a new piece of DNA. Okay, how we are going to cover this or understanding of these technologies is that there is no aspect of notes or dictation which I am going to be giving you here, but I will be writing the different steps in detail on the board and there will be slides. You have these slides. Okay, so as I am writing, please make sure you are copying the different ideas, right? We'll write the steps to and the different aspects of recombinant DNA technology. Basic idea wahi hai which I have shown you in the picture, that you take one part of gene, one piece of gene from one place, add another piece to it and you had a new gene. That's the basic idea. We'll see the finer aspects of that. Then we will do an example also which will make more sense to you as to why are we actually trying to do it. So there is a gene which we are basically interested in. So we first will have to isolate the gene. Isolation of desired gene. Let me take you to the example also simultaneously. So I am a pharmaceutical company and what I have decided is that I am going to sell human growth hormone. You might be familiar that a lot of people use supplements of human growth hormone for muscle building and other such aspects, or maybe let's just consider a normal condition where human growth hormone is deficient in a person. So as a pharmaceutical company, my goal here is to produce human growth hormone. What will I do is, I will take a human cell and the first step which I want to do is find out the gene which is producing human growth hormone and isolate that gene. If I want to produce human growth hormone, my idea will be to first get the gene. The gene for human growth hormone. The gene which is responsible for production of human growth hormone, I'm going to take that gene and I'm going to isolate that. Okay, that gene becomes my desired gene. Okay. For doing this process, we have different enzymes. Your cells have enzymes for cutting DNA. Isolate कैसे करोगे? Cut करेगा वो out of that whole DNA structure. You'll have to cut that part. And also later on when you need to join, so we have enzymes for joining DNA also. These enzymes have specific names. Cutting DNA enzyme is called nuclease. Joining DNA enzyme is called ligase. Nuclease and ligase. So nuclease के साथ you always get to read many other terms also. Restriction nuclease, endonuclease, exonuclease. So without getting confused, जहाँ पे भी word nuclease use हुआ, you have to remember that this is a DNA cutting enzyme. At which place it will cut DNA, based on that you can have some prefixes to that. But nevertheless, ground line यही. Nuclease means an enzyme for cutting DNA. Ligase means an enzyme for joining DNA. Okay. Now, if we have to produce human growth hormone, just that one gene alone cannot do that. It needs to be a part of a complete DNA structure. Okay, it needs to be placed in another piece. Just on its own, a single gene will not be able to produce the effect. So we need another piece of DNA to carry this desired gene. Okay, that piece can be, like in this example, taken from the bacteria. So the bacteria, along with their normal genetic material, they have another circular piece called plasmid. Okay, we have another piece called plasmid, and this plasmid can be removed and it can be cut open to make sure that there is place for the desired gene to come. This piece of DNA, it has no specific role other than to carry the gene. So the term which we use for this piece of DNA is vector. This is not the vector in diseases. This is the vector DNA for holding the desired gene. DNA used to hold the desired gene. Vector DNA is the DNA which is used to hold the desired gene. Most common examples of vectors are plasmids. So these can be obtained from bacteria, these can be obtained from viruses also. Here we are talking about bacterial plasmid. It can be obtained from virus also, but remember that. Independent circular DNA found in bacteria and virus. Independent circular DNA found in bacteria or virus, they can be extracted, they can be removed from the bacteria, cut, and we can add the desired gene with the help of ligase enzyme. And the new piece of DNA which will be formed would be the recombinant DNA. So we have the plasmid from here, the desired gene, and we can join both of these with the help of ligase enzyme and we get the recombinant DNA. Okay, so that you can write as your next step. Recombinant DNA formed by joining the desired gene to the vector. Now, once we have done this, okay, going back to our example, once we have added this to the added the human growth hormone gene to the bacteria, we want to actually do a production. Okay, and that production can be done by inserting this recombinant DNA back into the bacteria. Okay, we'll put it back into the bacteria, and now your bacteria has a gene for human growth hormone. So we can take that into the lab, we can make many multiple copies of it, and from those copies we can get human growth hormone. The bacteria, because they have the gene for human growth hormone, they will start producing human growth hormone. I can collect it, I can process it, and I can sell it in the market. That's the idea of human growth recombinant DNA technology. Okay. You take a cell, from that cell you first identify which gene you need. You cut that gene. Now you need another piece of DNA to hold this gene you are interested in, and bacteria and viruses provide this very wonderful aspect of plasmid. Okay, so that plasmid which we take, we remove it, we cut it, and we add the desired gene into this vector. Joining of DNA from two different sources gives us a new DNA called recombinant DNA. Because we said that universally the chemical nature of DNA is same everywhere, so it is possible for us to add DNA from different sources. We need ligase enzyme for joining and nuclease for cutting. This is then added to the bacteria so that bacteria can be producing the multiple copies of this bacteria will be produced, and commercially we can have this bacteria giving us human growth hormone which we can take and which we can add. Okay. See, this technology has very widespread applications in many areas. So any product of the living system which we want to produce, we can easily do this by replicating this process. Okay, whatever enzyme, protein, anything which we want to produce, we can actually do this with the help of recombinant DNA technology. Viral vectors have a very big application in vaccines also. The same idea of taking a viral vector, the viral plasmid, and adding another desired gene into it and giving it in the form of a vaccine. Okay, yesterday we did that Zydus Cadila vaccine, the DNA vaccine which was there, ZyCoV-D. This is a plasmid based vaccine only. तो जो भी character हमको चाहिए, whatever we need, we can very easily achieve that by combining DNA from different sources. Okay, and vaccination, because that's like the recent topic which is there, so it will make sense to understand it through this process. Recombinant vaccines as such is a standard term also. So this will again, all this will come under nucleic acid vaccines only. Recombinant vaccine का एक और example, one of the earliest example. So this is how it is produced in US, very famous hepatitis B. So, इसमें we have created a combination of this virus and we have put it in yeast. So, yeast is a fungus. So, the antigen part or the gene part for which or against which we need an immune response. I hope you have not forgotten the basic idea of DNA and RNA vaccine which we did yesterday. So that part, the antigenic part, that is taken, that gene, the gene which will help in production of the antigen, that is taken, it is combined with yeast, and then this yeast will express this gene and it will be given in the form of a vaccine and the person will be immune to that or develop immunity or develop immune memory to that disease. So, applications of recombinant DNA technology. You also have the more common examples of Covishield, Sputnik 5. They are also they are viral vector vaccines. What basically happens, let me tell you a little bit because I think a lot of you would have been vaccinated with Covishield. So I'll tell you a little bit about this. Covishield is a vaccine for COVID-19. What we do is from COVID-19, you will extract the gene. You will take the gene. Which gene? Gene which is responsible for production of antigen. Ye basic concept, I am sure by now you are familiar. We need the antigen. That is what we need to give in the form of a vaccine. Okay. So, वो antigen के लिए we can directly take a protein, we can take mRNA, we can take DNA. That's what we discussed yesterday. So, इसमें what we are doing, we are taking this gene and we are adding this, the viral vector which is going to be, the virus which is going to be carrying this gene is adenovirus. Why we are using this form of taking another virus and not COVID-19 exactly is because this virus, we find out a virus which in itself does not cause the disease. Okay, so your body, your human body has no immunity, no sensitivity to this virus. That means it will not respond to this virus. We have found out a virus which in itself is not going to be harmful. So the adenovirus which we extracted for Covishield, that was the virus which we got from a chimpanzee. Okay, so chimpanzee may it might be causing the disease, but human beings, human bodies do not respond to that. So हमने adenovirus ले लिया. Adenovirus में through recombinant DNA technology, we have added this gene. So this becomes the vector for holding my gene. And after this, what is done is, this virus is changed into a live attenuated form. It is weakened, it is alive, and then it is given in the form of a vaccine. So if we have to categorize Covishield, it's a live attenuated vaccine, but it is also a viral vector vaccine. We are not using the COVID-19 virus, but we are using another virus which acts as a vector for our antigen or the gene which is supposed to produce the antigen. And then the live attenuated vaccine, virus will do its job like normally it is supposed to. Okay. I hope this is clear. Take this one question, UPSC previous year's question. I did make you write what is a DNA vaccine yesterday. So don't ask me to define that again. Please look back. ऐसे पीछे वाला भूल नहीं जाना है. For genetics, it's very important that for you to be able to connect two topics, you need to know and you need to revise. Okay, the answer which I'm getting is D. Recombinant DNA technology, genetic engineering, allows genes to be transferred, 1, across different species of plants, 2, from animals to plants, 3, from microorganisms to higher organisms. Okay, and of course your answer is absolutely correct. It is going to be D, because technically the nature of DNA is the same. Its composition is same, universally it is the same. So you can transfer DNA from one place to the other. Okay. And sometimes, when we read things like animals to plant, then we kind of find it weird and we start second guessing ourselves. Yaar, animals to plant kaise jayega? Kyun jayega? Right? That's not for you to assess, right? In terms of technology, we might not have applications very wide ranging to this, right? But it can be done. In terms of genetic engineering, it can very well be done. It has been done. It has been done. Okay, so answer is D, 1, 2, and 3. Okay, let's talk about further aspects of this. We will see gene editing with this, but before we go into that, let's take a break and then we'll come back. Why are there two forms of DNA in bacteria and viruses, and one plasmid, another in the nucleoid region? Okay, this is a good observation you've made, Sunita, right? See, there can be multiple areas having DNA. If you think of the human cell or the normal eukaryotic cell also, in that also we have pieces of DNA other than our main DNA. If you remember, we had the mitochondria. We'll talk about the mitochondria in some time again. So that has an additional piece of DNA. The plants have plastids, which have additional piece of DNA. So it is very much a normal condition. So just like other cells, the bacteria and viruses can also have more than one place where DNA is found. Obviously, the secondary DNA or the second part usually is very small as compared to the main DNA, but nevertheless, that can be possible. Now, developing further into this idea of recombinant DNA, we have spoken about giving growth hormone to a person who we said maybe did not produce growth hormone normally. Okay, that's the idea which we have explored here. Now, think of the fact if this was a genetic abnormality. Okay. The reason why somebody does not have human growth hormone is because the gene for producing human growth hormone is not there or is not working. So instead of giving him human growth hormone, what if we have the aspect of giving the person the gene itself? Okay, what if instead of giving the product, we give the gene which is producing that particular product and then let the gene do its job? Sunita has asked another question. Can you explain the difference between recombinant DNA vaccine and subunit vaccine? So, recombinant DNA vaccine is the basis is DNA. आप उसमें DNA यूज करके वैक्सीन बना रहे हो. सब यूनिट वैक्सीन में आप प्रोटीन या और जो भी कंपोनेंट है वो दे रहे हो डायरेक्ट. इट्स अ न्यूक्लिक एसिड. द डेरिवेटिव रूट is the nucleic acid and here the derivative root is the product, the subunit, the antigen itself which you are going to give. That's the difference. दोनों में सेम कॉम्बिनेशन भी हो सकता है. सब यूनिट डीएनए वैक्सीन भी हो सकती है कि आप प्रोटीन बनाना चाहते हो, बट टू मेक दैट प्रोटीन यू विल फर्स्ट यूज अ रिकॉम्बिनेंट डीएनए. ओके, दैट कैन ऑल्सो हैपन. Right, you ultimately in the vaccine you will give the protein, but that production of that protein can be done using a recombinant DNA technology, right? Like the way we were producing human growth hormone, we could produce the antigenic protein also in that pattern using a plasmid, using a recombination. So recombinant technology पहले use करी and then subunit finally दिया आपने vaccine. That can happen. आप टर्म्स को डिस्टिंक्टली अंडरस्टैंड करो. अंडरस्टैंड व्हाट इज द मीनिंग ऑफ रिकॉम्बिनेंट डीएनए, अंडरस्टैंड व्हाट इज द मीनिंग ऑफ सबयूनिट, एंड देन इवन व्हेन दे आर कनेक्टेड यू विल बी एबल टू मेक सेंस. Okay, all right. So what I was talking about is, what if we can give the gene itself? Okay, we can give the person the gene itself which is defective. There are two ways in which we can do it. One is gene therapy. Please listen to this carefully. Gene therapy mein what is there happening? There is a non-functional gene. There is a non-functional gene which is present in the person, and to replace the function of this non-functional gene, ये gene है काम नहीं करे, so maybe there is a gene which is non-functional which is causing sickle cell anemia. Because of the presence of this defective gene, a person has sickle cell anemia. Now, to compensate for this, we can give a person with an additional functional gene. The way we will do this or give this additional functional gene would be normally through a plasmid. Okay, but here the process is not important here, understanding the concept is important. So if I have given an additional functional gene, ये non-functional gene तो है ही, ठीक है, it's doing nothing, it's present there. But additionally I have given a person a normal functional gene so that that normal function can be replaced. There is a gene which is supposed to produce human growth hormone. It's not doing its job. It's not doing anything. I've given an additional gene which will now produce human growth hormone. It will make copies and we will have the person being cured of the disease, gene therapy. It's a very expensive process, but it is being done for various diseases around the world. Okay, if you want, you can just write this in one line, gene therapy. A normal corrective gene can be added to take over to take over or to compensate. Compensate is a better word. To compensate for the function of the non-functional gene. Now there is another aspect which is further advanced than this, and that is gene editing. In this, we are not editing anything, we are just additionally giving one more gene. But we can also do editing. आप DNA ले लो, you take the chromosome where that defective gene is there, you cut it from that place and you add another gene. So by gene editing, we can add, delete, and also replace genes with the help of genetic engineering. Now we are going to learn how we can possibly do that. The basic ideas of recombinant DNA technology, which you have studied, that is what is going to be used. Okay. Let's see this most common technology procedure which is used for gene editing. I hope you have written this down. So, this is the cell. Inside the cell, there is a nucleus. Okay. And in that nucleus, we know that our genetic material is found in the form of chromosomes. Okay. We have the cell, the nucleus, and the genetic material in the form of chromosomes. Now, there is a part of this chromosome which is defective or which we want to remove or change. So what we will do is, we will first have to identify this part. For that, we can have different mechanisms to reach it or different mechanisms to guide it. So you can see here a guide molecule is coming. It will take us to this place, the exact place which we are interested in going. And we have technology today to program this for any gene. We can prepare guidance molecules for any gene which we are interested in. Right? So this guidance molecules takes us there, and then we get a DNA cutting enzyme, the nuclease enzyme which we have studied. So enzyme hai, guidance molecule hai, both the things are together and they will help us to remove or cut the DNA at that specific place. So we can just cut and remove it, right? The cells will normally, the cellular repair mechanisms will heal it, give us the normal DNA. Or if you want to add another gene, once this place is open, we can add another gene also. So that means we have replaced, we've removed a part, we've added a part. Okay. This process is very much possible. Last year, this technology actually won the Nobel Prize for Chemistry also. We'll talk about the specific details of this technology. I hope you have understood the basic idea. Cell hai, cell mein nucleus hai, chromosome hai. In that chromosome, in that DNA, there is one part, one gene which we need to alter. So we have a guidance molecule taking us to that place. We have a DNA cutting enzyme cutting at that specific place, and then we'll have the healthy DNA strand restored. Okay, let's talk about the specific technology, which is CRISPR, and then we will go into some more aspects. So CRISPR won the Nobel Prize in Chemistry last year, 2020. Its full form is Clustered Regularly Interspaced Short Palindromic Repeats. So because of this having a very large name, it's normally called CRISPR. It is also called molecular scissors, or in the common language, genetic or gene editing tool. Molecular scissor or the gene editing tool. Two components of it. First we said is a guide molecule. The guide molecule here is an RNA. Single guided RNA is its name. We also abbreviate it as sgRNA. We can prepare different types of single guided RNA depending on which gene we are interested in. Okay, which gene we want to alter, based on that we have the capacity to program this single guided RNA. Okay, this is the guidance molecule. This will take us to that specific place. Okay, and then you have the DNA cutting enzyme, Cas9. It is called Cas9 enzyme or Cas9 protein, because you know all enzymes are proteins. Basically, it is a nuclease. This is nothing but a nuclease. So you can maybe just write down in one line. The single guided RNA, guides CRISPR to specific sites, on the DNA, specific sites on the DNA and Cas9 protein cuts DNA at that specific site. All right. So see, this mechanism of editing of genes, this is a very effective one because of the smooth access we have from the single guided RNA. We can program it to become any type, any form. That is why this process is very, very advantageous. Okay. Now, Yash has asked a very interesting question that says that if we are editing a gene in one cell, right, what happens to the other cell? So for any process which we have to use, where we have to do a genetic modification like gene editing, that would be done at the stage of the embryo. Okay, your concern is very valid that there are so many cells. It cannot be done in an adult. It has to be done in an embryo where the number of cells are less. Okay, when the number of cells are less, so because we want to ensure that change is there in all the cells, it's reflected in all the cells, so that can be done only when we do it at the early stages of life. It can be done if you already know that there is a disease. Suppose there is a person who has sickle cell anemia. So you know that in any case, this person is going to give it to the next generation, right? That defective gene will be transferred. So in that case, we can actually edit his gametes also. You can edit the egg or sperm before it goes in for fertilization. So that can also be done. Or after zygote or the embryo, it starts to form, that has to be done at that stage only. Okay. Hari has asked, why can't we use DNA editing to cure Down syndrome and other genetic abnormalities? We can alter, or we can use it for genetic abnormalities, gene abnormalities, but not Down syndrome. This is what I have explained earlier also. There was a question कि पूरा chromosome ही जहाँ पे extra है. I did explain this, Hari, that when you have an extra chromosome or a missing chromosome, that kind of an abnormality, as of now, we do not know how to manage it. लेकिन अगर gene involved है, single gene, for that we have the technology. We have two technologies which we have studied here. One is gene therapy, and gene editing. Gene editing में आप एक्चुअली किसी का डीएनए में चेंज कर रहे हो. ओके, हाउ दीज टू आर डिफरेंट? In gene editing, you are actually manipulating somebody's chromosome. Okay. In gene therapy, we are not managing somebody's chromosome. We are not altering the chromosome which is already there, or the DNA setup which is there. We are just additionally giving one more thing. तो वो DNA structure के अंदर नहीं जाएगा. That will be kept sideways in its own plasmid in the cells. और वो replicate करके it will keep releasing the product which is there. Okay, so that's just introduction of a new gene, but not exactly into the DNA, but as a separate piece, this will be present in the cell. As of today, we do gene therapy, but we do not do gene editing. Gene editing is more on the experimental stage. Gene editing is something which we are still considering, that what it can be its impacts, what can be its negative impact. क्योंकि आप ये देखो, this has so much potential for misuse. I'm talking about treating sickle cell anemia with the help of editing of a gene, or treating deficiency of human growth hormone. But what stops me from giving it or doing it for any other type of alteration? Nothing stops me from doing it. Suppose I decide that I like babies who have blue eyes. Okay. No medical reason, nothing, but that's just something I like. So I can do gene editing into the embryo of my baby and I can give him blue eyes. I can add genes for blue eyes and remove the genes whatever he had, and I can produce a baby. Technically, it is very much possible, but is it needed? Should it be done? There are a lot of ethical, moral questions based on that. This is a concept called designer baby. Customized features in babies. Okay, this is a big concern. And we do not know how this could be used. So, CRISPR also in itself, there is a moratorium on its use, right? Gene therapy we do for various diseases, but gene editing, we are still waiting to see how it can be applied. If you are interested, there is this lady, Jennifer Doudna. Okay, Jennifer, या CRISPR सिर्फ आप याद रख सकते हो, and she has a TED talk. There's a 15-minute TED talk on CRISPR. So you can go to TED Talks and search CRISPR, right? And you can hear what she says. This is the first time 2 women scientists have won the Nobel Prize for Science. That is a first in Nobel Prize history that 2 women have won the Nobel Prize for science. Okay, it happened last year. But nevertheless, if you find this technology interesting, you can listen to what she has to say. She explains how they found out CRISPR, how it works, and also what are the concerns. Right? And China is trying it in military personnel. Yes, that's very correct, Shubham. Actually, nobody knows what China is doing, but there are definitely reports that they are doing military level manipulations where they are adding genes or creating variations which are favorable for the army. So maybe, better muscle mass, better agility, such features, right? Isn't it the individual's choice? It is definitely an individual's choice, but that's the moral ethical question here. Should it be an individual's choice? Because this manipulation is manipulation with the whole human race and the common gene pool which we have as a species. Okay. So this is an entirely different discussion, not really something which I want to go into now, right? We'll talk about these aspects also. Let's do a little bit more on the technology aspect. I want to talk about one process before I go further, that is IVF. So IVF is in vitro fertilization. I'm sure you've heard about IVF. What basically happens is there is an egg, there is a sperm, normal reproductive process. They're supposed to join and a zygote is formed. This zygote then divides into an embryo, and this embryo will ultimately be born as a baby. It will develop after the normal gestation period and it will develop as a baby. Okay. This process normally in nature, it is supposed to happen in the female body. This process of fertilization that the egg meets the sperm, this will happen inside the female body. Okay. But sometimes because of any medical conditions, any abnormality with the male, with the female, this process does not happen naturally. There are some challenges to it because of which naturally this cannot happen. So we have the technology to easily do this in the lab. Okay, we can easily do this in the lab. How? We can take the egg, we can take the sperm. We have a medical professional can remove the egg. Sperm retrieval is easier. So they are both brought into the lab, given the most ideal conditions of temperature, humidity, nutrients, hormones, whatever is needed. And in the lab, we can form the zygote. So once this process takes place in the zygote, we are going to call it in vitro fertilization. The process of formation of this zygote or the process of fertilization taking place in the lab, that is called in vitro fertilization. Now, after the zygote is formed, in the lab only we'll start the embryo formation. But the embryo as such on its own cannot be grown in the lab into a baby. Okay, for this, the embryo has to go into a female because she is the one who will ultimately give nourishment and the baby would be born. Okay. So, at the embryo stage, what we do is, this is transplanted into the uterus. It is transplanted back into the female uterus and then the baby is born after completion of that normal gestation period of 9 months. Okay, so this is the in vitro fertilization or the test tube baby. Now the baby will not come out of or be born in the test tube. Please remember that. It will be born from the female only its conception, the fertilization part, that has taken place in the test tube or in the lab. One more aspect which is in news, which the government is discussing, we have bills being passed on it, that is the idea of surrogacy. I'm sure you have heard this and you know what it means, but just to correlate it with this concept, once the embryo is formed, it can be transplanted in this same female who is the biological mother, who has given the egg, who has given the DNA, or if there is some medical problem, some reason why she cannot hold the baby, this can be transplanted into another female. When this happens, when it is somebody else's baby, somebody else's egg, somebody else's sperm, but a third person is brought in at the stage of transplantation of embryo, that person will be the surrogate. This surrogate has nothing to do biologically with the baby. It doesn't share genetic material. The only job of a surrogate is to provide nutrition and give birth. So the baby might be born through her, but she has nothing to do genetically with the baby. She is not the parent. Okay. And there are so many movies also these days which are coming on this concept. Like there's this movie Mimi. It was a real bad one, but if somebody has seen it. So in that, there is an Indian girl who becomes a surrogate to, I think, an American couple, and the baby which is born is American. Obviously, because all the characters of the baby would be determined by his DNA. And that DNA is coming from the egg and the sperm. Okay. The surrogate will give birth, but that process is only for providing the nutrition to the baby. Right? This transplantation, IVF में normally आप इसी mother को भी दे सकते हो. But if we don't give it to this mother, we give it to somebody else, we say it is a surrogate. And the government has a surrogacy bill. They are discussing about who should be allowed to become a surrogate, who should not be allowed, how it should be done, what should be the commercial angle in that, what is the legal status. Because India has a very big surrogate industry. We have towns, we have villages in Gujarat, which are solely dependent on surrogacy as a means of livelihood. So very big issue for India, because it is cheaper to hire a surrogate in India. A lot of people from all over the world come to India for this. Right? So you should be in touch with that bill also, you should have an idea as to what the government is saying, what is the logic, what are the different socio-economic, political aspects to that. That's a news which you will have to follow. Here I'm just explaining the basic concept. What is surrogacy? What is IVF? I hope this is clear so that we can go into our next topic. So this is the next topic which we are going to do. Can you tell me what do you know about this? Anything you know about Dolly the sheep? Clone. Yes, this is the clone. Dolly the sheep is the first mammal clone. Okay, the first mammal who was cloned, that was Dolly. And we're going to see what is cloning, how it is done, why it is done. See, for all the technologies which we will be doing, right, like recombinant DNA technology, like now we're coming to cloning or gene editing, whenever we are talking about a particular technology, we'll study the science of it here in the class, right? But after that, you can also look up and add the advantages, disadvantages, and applications. Advantages, disadvantages, and applications, in particular if there are any developments in India. Any developments which have taken place in India on that particular technology, you need to prepare them on this line. Okay. Okay. Now, talking about animal cloning, well, let's see what that is. Producing a clone means producing a copy, an exact copy, a Xerox copy of a particular animal. If we are going to make, that would be animal cloning. So, in this case, because we are making a copy, so technically there is going to be a single parent. There's going to be one parent, one person who is going to be copied and a new organism is going to be made. Okay, a new organism is going to be made. That is what is animal cloning. Now, how we go about that? For that, let's first just, for a minute, look at this idea of reproduction. So, you have 23 chromosomes coming from the egg, 23 from the sperm, and you have 46 in the zygote. So, the baseline is that for zygote is the first diploid cell, the first cell of your life, and then it will make copies, form the embryo. All of these cells would have 46. So, basically you need 2n number of chromosomes. For the zygote or for embryo to be formed, the organism needs a zygote. Now, I am telling you I want to use only one parent. We want to have only a single parent. So, if I want to have a single parent, suppose this mother, I want her to be the only parent. I want to create an exact copy of this mother. So, if I take her egg, the egg has only 23. I would need a place where I would need the other 23 also. Okay, 46 chromosome to chahiye zygote ke liye, life ke liye. So, what do I do is, instead of using her egg, I use the somatic cell. Somatic cells जो होते हैं, they would have 46 chromosomes. Somatic cells are all the cells of your body other than the gametes, other than the sex cells. Okay, so all the cells other than the sex cells are somatic cells, and because we want 46 chromosomes, so they are present only in somatic cells. So in this process, we will be using the somatic cells only. We will use a somatic cell. Somatic cell which is supposed to have 2n number of chromosomes. Okay. Now, this property of the zygote to start dividing into an embryo, okay, this property is because of the egg. The egg has an apparatus to start developing into an embryo. The egg has the apparatus which will ultimately ensure that how nutrition will be taken, how placenta will be formed, everything which is needed for growing of the embryo, that whole information is in the egg. Okay, so that means the egg we need. It's in the cytoplasm of the egg. So we need an egg. Just taking a somatic cell, that means the cell of your hair, the cell of your skin, it can be sufficient to get us the DNA or the 46 chromosome, but it doesn't have that property that it will change into the embryo. Okay, तो अगर हमें embryo बनाना है, for that we will need an egg. Egg के बिना नहीं बन सकता. Okay, so now what we are going to do is we are going to take another organism and we will get an egg. So egg है n number of chromosomes. But we do not need the egg's chromosome. Because chromosome we have decided we will take from this person who is giving the somatic cell, whose copy we are trying to produce. So what we do is we do a process called enucleation. Enucleation means removal of egg. Enucleation is the removal of the egg which is there. Removal of the nucleus. So we will remove the nucleus in both of these cases. What happens then? You have the egg. We have removed the nucleus from the egg. It is now empty. Why we have removed the nucleus is because we do not need the chromosomes coming from the egg. We only need the chromosomes from the somatic cell or from the animal we are cloning. Now, in this case of a somatic cell, we will keep only the nucleus. So, this is somatic cell nucleus. And now what we are going to do is we are going to add this nucleus to the egg. Okay, so we'll take this nucleus and we will put this in the empty egg. So this is now egg with somatic cell nucleus. Egg with somatic cell nucleus. And we can stimulate this in the lab. Stimulate means give it all the necessary inputs which are needed. Stimulate in the lab to form embryo. And this embryo then can be transplanted in the uterus of a reproductively active, reproductively healthy female. Let me take you through this example again, with the help of an example. Suppose I decide that I am going to create another Amitabh Bachchan. I want to clone Amitabh Bachchan, and for that what I would need is, I would first need a somatic cell from Amitabh Bachchan. If I have to clone him, I need a somatic cell. That means any cell other than his sperm, other than the sex cell, other than the gamete, any other cell from his body I will take, and that will be my somatic cell. Now, I have to find another female who will give me the egg. Because I need the egg, because without the egg, the embryo will not be formed. So, I can ask somebody to donate an egg. So, I'll ask Kareena Kapoor, please give us one of your viable eggs. Okay. Now, her DNA we do not want. We just want the cytoplasm and all the things, other things in the egg. So, we do not want her chromosomes. So, we will do enucleation. We will remove her nucleus and we will remove the nucleus of Amitabh Bachchan's somatic cell. So, we have Amitabh Bachchan's nucleus with all his 46 chromosomes. And we have an empty egg from Kareena Kapoor. Now, we'll fuse this. This process of transfer is called somatic cell nuclear transfer. Somatic cell nuclear transfer. Okay. And now I have an egg with 46 chromosomes, and that's the requirement, that's the basis of the zygote. The zygote is having 46 chromosomes, so that is the stage where embryo can be formed. So with the egg containing 46 chromosomes, we can treat it in the lab. It will start forming the embryo under the most ideal conditions, and now we can give this to a female. So we can give it to any female who will ultimately give birth. So maybe we can ask Vidya Balan that if she can hold this embryo, then she will give birth to a baby. All the properties of that baby, everything from his height to his hair to his voice, everything will be like Amitabh. Of course, there's going to be a difference in the age, right? The baby will not be born 6 feet tall and with a beard. That's not going to happen. It's going to be a baby. But because all the chromosomes, all the genes are exactly picked up from Amitabh Bachchan, so this baby would be having everything, all the credentials like that of Amitabh. That is what is cloning. Okay, that's what is cloning. Making an exact copy of an organism. I'll show you this same thing about Dolly also, how Dolly was made. So this is the original sheep which was used to make Dolly. See, any animals which we find that to have, favorable characteristics, okay, maybe she's giving very good wool or anything like that. So we can make more copies of that organism. That's one idea or one aspect of the use of cloning. So we have a sheep, we take a somatic cell. In this case, we took the udder cell. Udder cell was the cell from the milk gland, the milk glands of this sheep. So we took a cell, and from that somatic cell which we took, we only needed the nucleus. So we've taken the nucleus because we wanted her 2n. Okay, we wanted her a 2n, that is her complete set of chromosomes. Then we take another female who will give us the egg, because we need the egg, but the egg will be enucleated, and then we'll put this donor's nucleus into a new cell, a new egg is formed with the complete set of chromosomes from the sheep we are trying to clone, and we'll stimulate it. We'll have more than one cell, an embryo will be formed, and then we'll put this embryo back into a female to give birth to a lamb, and this lamb is the cloned lamb, which is the exact copy of the person or of the sheep which gave the somatic cell. Okay, understood? Okay, Deepshika has asked, can we not use the egg of the parent for cloning purpose instead of the donor? Yes, if it is a female, we can use. Okay, that's very much possible, but just to give an distinction, because it could be a male also, and the male will not have the egg. So that's why we have taken two different organisms, right? Amitabh Bachchan doesn't have an egg. So otherwise, if he had, we could very well have done it, right? तीनों चीजें same organism भी कर सकता है. Like if you want to clone Kareena Kapoor, you can take her somatic cell, you can take her egg, and after the embryo is formed, you can actually put it in her body and she can give birth. So no second person is needed. There is no problem in that. This is just to make you understand of the different steps. That's why it has been distinguished like this. Okay. Do you understand this? If किसी का बेबी नहीं होता तो ट्रांसप्लांट के बाद यूट्रस में हो जाएगा. See, the defect, Mumtaz, might not be, if you're talking about surrogacy, so the defect might not be in the uterus, right? The defect might be in the sperm, which has low motility and it is not able to reach inside, right? So it's not necessarily that IVF is supposed to be done for a female defect. The second thing is that nowadays people are having babies without having a partner, right? If I decide that I want to have a baby alone, okay, so I do not have a partner, but I want to become a single mother. Now, what are my options? My options are to get a donor. Have you seen that movie Vicky Donor? Right? In that, there are couples who are not able to conceive normally, so they get a donor. So a lot of people are becoming single parents also in that sense. So if I want to be a single parent, I am reproductively healthy, everything is fine, but I do not have a partner, or I do not want to have a baby with a partner, so I can get an artificial process of the IVF done with the help of a donor. There are many permutation and combinations, of course. because of which or how it is going to happen. So it's not just one thing. Uterus is different. You have tubes which can be blocked, but the uterus can be fine. So this is a complicated idea for you to give such a simplistic answer. Unable to get difference between surrogacy and cloning. ये जो है, the person, the female who will be giving birth in this part also, like Vidya Balan who gave birth to Amitabh Bachchan's baby, that person is a surrogate only. ठीक है? Because biologically इसका इस clone से कोई लेना देना नहीं, right? But in cloning, the basic fundamental which is different is that there is only one parent. That's not how it is in surrogacy. Normal surrogacy जो हम करते हैं उसमें ये वाला प्रोसेस तो नॉर्मली है. देयर आर टू पेरेंट्स, देयर इज अ मदर, देयर इज अ फादर, देयर इज एन एग, देयर इज अ स्पर्म, राइट, एंड देन वी पुट इट इनटू समबडी एल्सेस बॉडी. तो वो तो सरोगेट रहेगा. लेकिन क्लोनिंग में देयर आर नो टू पेरेंट्स, देयर इज ओनली वन पेरेंट, one person giving all the genetic information, right? As of today, human cloning is not allowed. Okay, human cloning is not allowed because again we do not know how the misuse of that is going to happen. Of course, there are always reports which indicate that somewhere some human beings are being cloned, or when Osama bin Laden died, there was this report that the Navy SEALs did not find the real Osama bin Laden. There were clones. Osama bin Laden has 4 clones and they probably killed a clone and he's still hiding somewhere, maybe in Pakistan, who knows. But there were theories like that, right? But officially, legally, the status is that it is not allowed. Okay, the status is that it is not allowed. Human cloning cannot be done. Basic idea आपको cloning का यही याद रखना है, that there is one single parent. Okay, there is one single parent. One person will give all 46 chromosomes instead of 2 parents coming together and giving 23 chromosomes. ठीक है? Okay. Now I have 2 more topics to be done with you, actually 3, but I'm not able to decide if I should do it today or in the next class, because I wanted to finish today, but we are slightly off in our scheduling and we have half an hour more to go for the class. Okay, let's finish. We'll take some time extra and we'll finish today. The next technology, Mitochondrial replacement technology. Yeah. This is not a question for you to answer because I know every time this question is put, there's always going to be a debate. Today, next class. अगर next class भी मुझे लेना हुआ तो that will just be a 1 hour का class. So there's no point, wasting your time just for 1 hour. Right? So we'll try and finish it today only. Right? We don't have a lot of things left. So let's finish it. We spoke about having a single parent. Now this is a situation where we have two parents. Okay, this is a situation where we can have one extra parent. That means total of 3 parents. Right? So how is that going to happen and why is that going to happen? That's what we are going to see. Remember mitochondria, your favorite cell organelle? Its main property was that mitochondria had its own DNA, which is called mitochondrial DNA. So mitochondria has its own DNA, which is called the mitochondrial DNA. Okay. And the special property of this DNA is that it is inherited only from mother. All the mitochondria in your body are coming from the mother. Right? Like the normal DNA in your bodies, it is coming equally from both the parents. Mitochondrial DNA is coming only from the mother. All your mitochondria are from the mother. Now, the latest research shows that there is a possibility in having some defect in this DNA. This is a very small piece of DNA. It has only 37 genes, right? But still, if there's those small genes, see, as such, that less number of genes instead of the thousands of genes which you have otherwise, it won't make any difference. Lekin agar defective hai, then it can cause a disease. So there is a possibility of having a defective mitochondrial DNA which can cause various diseases. One example of which I'll give you is a disease called Leigh disease. There is research going on which is discussing on aspects of this mitochondrial DNA being involved with other diseases also, like Parkinson's, even diabetes. They say they can trace some link between the mitochondrial DNA. Okay. So in any case, once we have observed that there is a female, she has a defective mitochondrial DNA, she has some disease which she is suffering from, which is because of mitochondrial DNA. So we can ascertain one thing, that she will definitely pass it on to the next generation, because all her children are going to get her mitochondria and all of them will have the possibility of getting that disease. So in order to avoid that, what we can do is we can replace this defective DNA. Okay, we can replace this. We'll just now see how we can do that replacement. We call this mitochondrial replacement technology, or we can also, in common terms, it is called 3-parent baby. Please write all this. I shouldn't have to tell you that. I'm hoping you are writing. Or we say pronuclear transfer. Why we are using the word pronuclear transfer is we are not exactly talking about the nuclear DNA here. The DNA in the nucleus, that's not the one which is being questioned, which it is not the one which we are bothered about, but the DNA which we are bothered about or which we are talking about is the DNA outside the nucleus. So the term we are using for it is pronuclear DNA or pronuclear transfer. So these are the three names for this kind of replacement. We'll just see how it happens. But the basic idea you should know, we have DNA in our mitochondria, right? Then we are going to get that DNA only from the mother, and once we know the mother's DNA is defective, that means it will pass on to the child also. So we are going to replace it in some way. Okay, how we replace it? Let's see that. So this is the egg of the mother who is having defective DNA. So they have written unhealthy DNA. We can specify and write defective mitochondrial DNA. Please keep writing this one by one in the heading. You don't actually need to draw, but you can just write defective mitochondrial DNA in the mother's egg. Okay. Then now we don't want her mitochondria, so what we will do is we will bring another egg with healthy mitochondria, or we can write here normal mitochondrial DNA. So the mother has defective mitochondrial DNA. We want to replace that, and for replacing that we can bring in another egg which has a normal mitochondrial DNA. Okay, this same thing which we did earlier, we'll do that same concept. We will be doing enucleation. Enucleation means removal of nucleus. Okay, enucleation is removal of nucleus. So this nucleus can be removed, and in the second case of the donor egg, here also we'll remove the nucleus, but nucleus contains the main genetic material, so we don't want her nucleus of this donor egg. So we take the empty egg. Empty egg means the egg without the nucleus. And of course, egg without nucleus, but the egg with normal mitochondrial DNA. Now, we will do a fusion. We will join this. We will do this transfer. You can say pronuclear transfer. And we have the nucleus of the mother in the donor egg, which has healthy mitochondrial DNA. Okay. Now this egg can be fertilized with the help of a sperm, and we can have the zygote formed, then the embryo formed, then being transplanted. It's an application of things you have already done. Basic idea ye hai, there is a person who has defective mitochondrial DNA. We can identify that with the help of different tests. We can identify this once we know that the mother is suffering from a disease, right? And we'll take a donor with normal mitochondrial DNA, we'll remove the nucleus from that egg, and we can then put the nucleus into this. We'll have a mitochondria which are normal, and we have the egg. Just to give emphasis to the fact that DNA is coming from a third source. See, this DNA is not significant. I told you genes are very less, 37 genes. But just because this is the main DNA we are interested in, this is the DNA which we want to actually get, we also give the reference of a third parent baby. So this donor can be addressed as the third parent. However, genetically it will not reflect majorly because the number of genes are less. Okay, mitochondrial replacement, pronuclear transfer, or the three parent baby. One more concept is there that आप egg की जगह zygote में भी ये चीज कर सकते हो. Right? We are using the egg here, but instead of the egg, if we have a zygote, think about that. यहाँ पे एक zygote है, right? We can get another zygote. This is the zygote of the two parents. Fertilization already taken place. Mother's egg and father's sperm, they have been joined. So bring another zygote. उसमें से nucleus निकालो आप, and the same thing, adding that nucleus, same process could be done. क्योंकि basically तो जो main DNA है हमारा 2N, that is in the nucleus. So it can be the egg or it can be the zygote. It will not make much difference. The same idea of mitochondrial replacement for treating mitochondrial diseases is going to be applicable. See, all these individual portions, they are very important when it comes to questions in prelims. Mitochondrial replacement, already 3 questions have been asked. Gene editing, already 2 questions have been asked. On recombinant DNA technology, already 2-3 questions have been asked. On all of these, they will stay in news. So, आप current affairs जैसे जैसे पढ़ोगे, you'll read, keep reading about these things only. Nothing new is going to come there. It's just going to be application of these ideas or something, some development somewhere which has taken place. Okay, so make sure you have understood the concept properly. That's the basic line. That's the main point of significance. Next topic is stem cells. Next topic is stem cells. Please explain zygote example. Zygote कुछ नहीं है. What was the zygote? कहाँ गया हमारा zygote? ये रहा. ये बनाया ना हमने zygote. So we have the mitochondria in the zygote coming only from the egg. That's what we have said. Mitochondria come only from the mother. So we can take the egg or edit it, or we can join the egg and the sperm. It's just the steps how you want to do. आप egg को alter करो, बाद में sperm add करो, या आप sperm पहले ही add करो. It will not make a difference because the mitochondria in the zygote are all coming from the egg only. तो आप zygote बना के, एक और zygote बना के, donor के साथ भी zygote बना के, उसमें भी mitochondrial replacement कर सकते. Same idea. It's just that जो IVF का process है, जो fertilization है, that you have done before. That's the only point. Okay. All right. Now stem cells. Now there are some cells in the body which are going to be in a position where they are going to be able to divide, and they are also will be able to differentiate into different types. But then it will become four parent baby. Yeah, you can say four parent baby. Most of the times they do it with the egg also, but technically it can be done with the zygote. Yeah, it is absolutely legal. It is the procedure which is done. Okay, so we have different cells in the body, different cells which are supposed to be doing different jobs. Okay, different cells which are supposed to be doing different jobs, like you have muscle cell, you have hair cell, you have fat cell, like that. But that's not how originally life started. Okay, that's not how originally the system started. What happened is that your life started with the zygote. Okay, your life started with the zygote, and zygote is one cell. Right? Zygote is just one single cell. Now, from that one cell, your whole body has come. So, 2 points you have to consider here. Ek to ye, that the zygote was able to make more cells. Today, your body has billions of cells. Okay, your body has billions and millions of cells. So, because of that, that means that that one cell was able to make more copies, and from that one cell only different types have come out. Different types as in blood cells, muscle cell, hair cell, skin cell, fat cell, all of those cells have come ultimately from that one zygote only. Okay. So this property of one cell changing into more than one cells and having more properties, right? Not just zygote, after you are born also, today also, your body keeps producing new cells, right? If somebody gives blood donation, so blood will be replenished, right? You will have blood cells being formed again. That means there are cells which can form new cells and also increase the number of cells. This property of a cell is what is stem cell. Okay, this is the property of a stem cell. So you can just write this down. Just write down what is stem cell, then we'll go into its different aspects. Stem cells are unspecialized cells. Stem cells are unspecialized cells that can grow and divide into different types of cells. That can grow and divide into different types of cells. Okay. Now, let's talk about a few properties associated with your stem cells. The two terms which I'll write, one is pluripotent and the second one is multipotent. Pluripotent means there is one cell which can change into all cell types. one particular cell can change into all the different cell types. Because that means this is a stem cell which can change into muscle cell also, into fat cell also, into brain cell also. That property we will say is pluripotent. And multipotent will be one cell can change into one type of cell, not all. These are the two properties associated with stem cells. One, I'll give you examples also of this type. Pluripotent would be one cell is there, that cell changes only into one, can change into all the cell types. Okay, example यहाँ पे है embryonic cell. So the cells in the embryo in the early stages of life, they have the potential of changing into any type of cell. Okay, but after you have grown up, after you have been born, your body is in place. At that time, in adults, the stem cells are there, but they are tissue specific. What does that mean? You might have seen that your skin keeps shedding every 1 month, right? 28 days, 23 days, like that, there's some number, right? Or new blood cells would be formed, or say some organs are disintegrating and they would need to be replenished, would be formed again. For that, your body keeps a reserve of stem cells. But these cells, the adult stem cells, they are not specific to any type. ऐसा नहीं है कि जो brain में stem cell है उससे muscle भी बन जाएगा. The stem cells in the brain will only form new brain cells. The stem cells in your bone marrow, they will form blood cells. We've discussed blood cells are formed in the bone marrow. The stem cells in the liver will form new liver cells. So they are specific to one type which they will form, not to all the types. Okay, if I take a stem cell from your bone marrow, which is present in your adult life, you cannot change it into an adult, into a skin cell. वो blood cell ही बनाएगा. Okay, that is what we call multipotent, and the other type is the pluripotent. Okay, so you can write both these terms. Write down pluripotent first. Pluripotent. The stem cell can form any cell of the body. Pluripotent is a property where a stem cell can form any cell of the body. Example लिख सकते हो आप embryonic cell. Example is embryonic cell. And then multipotent, a stem cell can change into some particular type of cell, and some particular type of cell and not all the cells and not all the cell types. Based on this idea, we just summarize this here into types of stem cells. First type, embryonic. Please write this down. Types of stem cell. embryonic stem cells. Cells which are present at the early stages of life. Cells which are present at the early stages of life and can form all the cells of the body and can form all the types of cells of the body. Then next, adult stem cells. Adult stem cells. They are present in different tissues. They are present in different tissues in the body and can change into cells of that specific tissue. Can change into cells of that specific tissue. There is a third type also, which is called induced pluripotent stem cells. Induced pluripotent stem cells. This is something which we can artificially create in the lab. So what do? We take any normal cell, any cell like muscle cell, blood cell, any one cell I can take. I can treat it in the lab and make it pluripotent again. Okay, I can make it pluripotent again. That means I can make it undifferentiated and I can give it the potential to divide. So then the muscle cell which I had taken, once it is induced pluripotent stem cell, then I can change it into any other type. Okay, that muscle cell, after it's pluripotent, I can make it into a blood cell. Okay, write down induced pluripotent stem cell. Induced pluripotent stem cells. It can be induced. It can be induced to become undifferentiated, and develop the potential to divide. Develop the potential to divide. Okay. Now see this division ka concept. There are many more terms also just to distinguish between adult and embryonic stem cells. I gave you these terms. Totipotent, the term which you have used. Totipotent is used for the zygote. इसमें और pluripotent में difference क्या है? That along with making all the cells of the body, this zygote can also form extra embryonic membrane or extra embryonic tissues. So when the baby is developing, there are a lot of other structures also which are created, like you have the umbilical cord, you have the placenta for nutrition. Where is all that coming from? A normal pluripotent cell doesn't have the potential to do that. But a totipotent cell will be making all these, and the totipotent cell, only totipotent cell which we have is zygote. So it has the property of not only forming all the cells of the body but also forming the extra embryonic tissue. Okay, it is different from these two. In that case, the sperm will be from the same father. Achha. If you're talking about the zygote mitochondrial replacement, it doesn't matter the sperm is from the same father or from the different father, because the sperm का जो main content है वो nucleus में जाएगा. Sperm is very small, it has nothing but that small nucleus, a little bit of cytoplasm. So those 23 chromosomes are the main thing in the sperm, and that you will remove. As such, we can use the same sperm also, but even if that's not the case, the nucleus will be removed, it doesn't make a difference. Now I've told you about stem cells and about the idea that how they have a potential of forming other types of cells. So think about the applications. Where do you think this can be used? Where can stem cell technology be used? Where can we have their application? We have cells which will divide and form any type of cell. So answer me on that front. See, we have cells which can change into another type. Okay. Now, how do you get it and where do you get it? One way to get it is through embryonic stem cells. They are the best kind of stem cells. Why? Because they can be changed to any type. Okay, they can be changed into any type. So now we have started storing embryonic stem cells. Okay, we have now started storing them. That means earlier on, for you and me, it has not been done. Okay, how do we store it? At the time when the baby is born, there is a umbilical cord which is connecting the baby to the mother, which is cut. Okay, so you might have seen this in different movies that they show you cutting off the umbilical cord, right? That tube which is there. That tube contains some embryonic stem cells. Okay, that tube contains some embryonic stem cells. So we can take the cells from there and we can store it. It can be stored for 100 years or 50 years, however you want. We have the technology to do that storage now. Okay. This is the use of embryonic stem cells. Advantage क्या है कि embryonic stem cell लो, किसी भी form में change कर लो. But for everybody, embryonic stem cells are not preserved. So what are the options? The option is to use adult stem cells. अब adult stem cells का problem क्या है कि they are tissue specific. And extraction of adult stem cells is very difficult. You want to create brain cells, you have to go and extract it from the brain, which is very, very difficult. You have to extract it from the spinal cord. Okay, you have to extract it from somewhere in the skin. So because of its specificity, there is a challenge. As of now, हम adult stem cells ही use कर रहे हैं. We don't have a lot of other options, right? And that's what makes this technology also very, very exceptionally expensive. But if we have more and more samples of embryonic stem cells, if more and more people preserve it, you might have seen some advertisements also. There's this brand called LifeCell. It is endorsed by Aishwarya Rai, if you've seen those advertisements. So if embryonic stem cells are there, if they are available, we can easily do the treatment without having to worry about extraction. Okay. And the other option of having induced pluripotent cells, creating those cells, that can also be very helpful. क्योंकि उसको भी फिर लेके जिस मर्जी टाइप में बना लो. Right? Now the application part. Right? I have these potential applications in front of you. You can copy some of these. You have this slide also. You are mentioning some of the points. Replace damaged tissue, organ, right? In this replacement of tissues and organs, the very, very biggest advantage is that we can create new things. Anything which is damaged, anything which is destroyed, आपको orthopedic injuries हैं, there are skin burns in somebody, we have the potential of giving new cells. Okay, so almost everything they have covered here, brain, heart, muscle, pancreas, liver, skin, spinal cord, blood. Everywhere, wherever there are cells missing, like जैसे sickle cell anemia में red blood cells जो हैं, they are not properly formed. We can give new red blood cells with the help of stem cell therapy. Right? किसी को arthritis है, the bone tissue is disintegrating, so we can give new bone cells. There's a fracture somewhere and severe damage to bones. Multiple line fracture is there, so we can give new bone cells. That is the immense potential of stem cells. We can give new things, right? Transplantation you have mentioned. So organ transplants ke liye this is a blessing. Somebody needs a heart transplant. You create new heart tissue with the help of stem cells. Somebody needs a liver transplant. You create new liver tissue with the help of stem cells. The idea is to make new cells. We can make new cells very easily with the help of this technology. Okay. If we have embryonic cells, you know how these products are advertised, the products which sell embryonic cell, they advertise you in the form that not only for the person who has stored his embryonic cell, not only for that baby, but for anybody in the family. Because you know organ transplants also can be given within the family if there is a matching. So if those cells match, they can be used. So today a baby is born, after say 2 years, his grandmother develops Alzheimer's disease. She can find a cure of Alzheimer's disease by getting new brain cells with the use of the stem cells of her grandson. That's the massive aspect of these stem cells. Also, very, very important for research and development. आप research कर रहे हो, कोई new medicine है, कोई new technology है, you want to study the effect of some disease. Instead of doing it on pigs or other animals, we can use human blood, human stem cells and test it on them. The results will be more, you can say, significantly in line with the actual human body because we are using real human cells. So massive significance in research and development, checking for new medicines. You want to check the effect of some medicine. I'm making a medication for, say, acting on my blood cells. So I can take blood cells and see the impact directly. Okay, so research. Then regenerative medicine. The term we use is regenerative medicine. So regeneration of any of these parts of your body. Okay. As of now, the challenge is a lot is not understood about the negative effect. That what can go wrong. There is adult stem cell का एक challenge है extraction. मिलते ही नहीं है. Right? But other than that, when we actually give a stem cell treatment to somebody, what would be the impact of that? That is not very well understood as of now. Okay. Can we use stem cells of one in another body? Yes, we can use. We have a term for that also. Okay. So when we're using somebody else's stem cell, most of the times you have to use your somebody else's only. The term is allogenic. Allogenic means coming from another person. आप पूरे पूरे organ जब किसी और से ले सकते हो, right? Then why can't you take stem cells? We have technologies or ideas where you can get grafts from other animals also. Right? That's also recently it was in news that something from the pig has been taken, some organ. So definitely You have to do some management in the body in terms of some medicines which would be required, but that's very much possible. And India, maybe recently this has been achieved. Can the bone marrow be created? The bone marrow cannot be created as such. The bone marrow is there in place inside your bones. You don't have to actually create it. Can it cure cancer? It can, we won't say it can cure cancer, but it can definitely be a treatment of cancer. Think of a cancer where there is, say, one particular type of cell which are missing. ठीक है, कुछ अब नॉर्मल लेवल बॉडी में ऐसा हो गया कि there is one particular type of cells whose number is decreasing because some other cells are being produced more, like leukemia, blood cancer. उसमें white blood cells जो है, their number is less and some other cells have increased. So we can use stem cells to create that, to give those additional cells. It will obviously depend on different types of cancer, but this is very much possible. Okay. Let's do one question, then we'll go to the last part of our discussion. Cloning has nothing to do with stem cells. Cloning में we are using normal somatic cell of somebody's hair, somebody's body, like वो sheep में दिखाया था हमने, we were using the cells of milk glands, तो उसमें stem cells का कोई रोल नहीं है. Okay, so D, B, C also. With reference to stem cells frequently in news, which of the following are correct? So, first statement, stem cells can be derived from mammals only. No, this is absolutely incorrect. From all organisms, basic idea stem cell क्या है? Stem cell is an dividing undifferentiated cell. So it is not exclusive to mammals. So one is absolutely incorrect. Then second, it can be used for screening of new drugs. Screening of new drugs means testing new drugs, testing new medicine to see their effect. For that we can use stem cells. This is correct. Right? Then stem cells can be used for medical therapy. That is also correct. As a treatment option, it can very well be done. So, answer is B, 2 and 3 only. B, 2 and 3 only. Now, Pranika has asked me how are twins formed. There are two possibilities of twins forming. Normal process reproduction का क्या है? Egg है, sperm है, they will join, zygote will be formed. Okay, this is the normal process of reproduction. Sometimes what can happen is this zygote can make two copies. So, two zygotes will be formed from this one zygote, and this is how you will have twins. This one is the identical twins because they are having formed from the same zygote, they are copies. It's just that that zygote has created two copies. So they will exactly have the same genetic material, same DNA, everything is same. They are identical twins. Right? There is another possibility of non-identical twins. What are these? So, normally what happens in females that in 1 month, one egg is released. So, females have 2 ovaries and in every month, one of those ovaries turn by turn releases one egg. Sometimes in a rare condition, she can produce 2 eggs. Both the ovaries have released an egg, so 2 eggs would be formed. This is a rare situation, but it can happen. Now sperms are coming in millions. So if both these eggs get fertilized, we have two different zygotes, because the eggs and sperms are different. Genetically, these zygotes are different. So two zygotes are formed and you have non-identical twins. These same twins, in terms of their genetic correlation, they will be as similar as normal siblings. तो जितना correlation siblings में होता है, जितना linkage genetically siblings में होता है, born in different births, they'll have the same relationship, just that they are born together. But they are from two different eggs, two different sperms, so genetically they will be different. So when you have a boy and a girl, it is happening. The possibility of that happening is because of the idea of non-identical twins. Okay. The last part is just me giving you some ideas as to how to prepare that welfare part, right? We have completed with our technologies. We have completed with all the aspects of science which we needed to do. Okay. Now we are just going to talk about how you are going to build up your notes and how you are going to think about different aspects of the human applications. So biotechnology can be divided into 4, in fact 5 types. One more I will add here. So this division is based on their application in different fields. Okay, this is based on how we find application in different fields. So you have blue biotechnology for marine products, aquatic organisms, your seafood. Then you have green biotechnology for agriculture. That means when you are doing some kind of gene editing, some kind of engineering on the agricultural crop. Then red, medical. All the aspects which we have studied are medical only. Vaccine, recombinant DNA technology, stem cell technology, they are all medical applications. And you have white in the industry. So we might be producing some chemicals with the help of living systems, like maybe some enzymes we are using. I'll give you the most common example of use of enzymes. Your dishwashing bars. Okay, the dishwashing soaps which are there. So dishwashing when you're washing your utensils, one of the main things which you have to eliminate is oil. Now oil is a fat, right? I hope you have not forgotten that. So oil is a fat. So if we can have a fat digesting enzyme and add it to our soap, that enzyme will help in breaking down the fats and removing it easily from your utensils. So that application can be an application under white biotechnology. The last one is gray. Gray is for the environment. Environment there are two problems basically. You will read this in a lot of detail when you study environment. There are two problems. One is how to manage the waste and the second is to control pollution. For both of these, biotechnology can be used. Let me give you an example. So we have a group of genetically modified bacteria called oil zapper. So when the ships around the world, they are transporting oil from one continent to the other, there is always a possibility of a spillage, oil spills. But once these oil spills, they are very damaging to the water to the living organisms living in there, right? So that oil has to be removed. Clearing of that oil has to be done. Now, to clear that oil, there is this special genetically modified bacteria which is created, which is oil zapper. इसको आप पढ़ोगे डिटेल में एनवायरमेंट में. तो ऑयल जैपर का काम क्या है? वी हैव दीज बैक्टीरिया हु कैन डाइजेस्ट ऑयल. सो वेरेवर देयर इज एन ऑयल स्पिल यू कैन स्प्रे देम देयर एंड what will happen is that the oil will be removed because that has been digested. Okay, so this becomes an environmental application, right? A little bit I want to talk about agriculture. If you can tell me this, the question which I want to ask is, why would we want, or what do we want to achieve if we are doing some editing, some changes in our agriculture crops? हमारा objective क्या है? Why do we want to do that? That's my question to you. So when we make any kind of genetic changes in a particular organism, we can call them genetically modified organisms or GMOs. In crops, when we do it, and this is something which is always in news, you will have to track this. Once we are using it in agriculture, this concept of doing some editing, all the processes are going to be the same. Wahi gene editing, recombinant technology, right, which we will use for the purpose of doing some changes in your crop. So why would we want to do it? That's what I had asked you. Okay, Pinky says good quality and more production. Okay, that's the first point, relevant point. High yield, high productivity, more nutrients. Okay, this is also a good point. More nutrients, extra nutrients. For food crops, this can be a very good point. Then Abhishek says pest resistance. Again, very relevant. So, आप ये सब चीजें, you will read about them in the news. Now tell me, why do you think there are concerns? What do you think can be the problem? हम ये use करना चाहते हैं, we are getting some benefits. What can be the challenges? What can be the problems to this? Tell me that. So high yield के लिए, you have the example of GM mustard. Okay, ये सब current affairs में आएगा. More nutrients के लिए, we have an example of a special rice variety called golden rice, which additionally has vitamin A. तो एक rice variety है, उसको हमने recombinant DNA technology use करके we have put in vitamin A in that rice. So that rice variety is called golden rice. Then pest resistance के लिए we have BT cotton. We also have same technology से BT brinjal. As of today, in India, BT cotton is the only crop which is allowed for commercial cultivation. India में currently there is only one crop in which commercial cultivation is allowed. Only one genetically modified crop. Okay. Rest everything is trial. BT brinjal को भी recently हमने final stage trial में डाला है, but cultivation is not being done because of certain concerns. So what are the concerns? Okay, destroys soil retainability, soil pollution, problems of unknown side effects, prevent soil degradation, environment, not natural, artificial soil degradation, productivity, okay, reduce soil nutrition levels. Okay, I'll give you broad headings. Okay, you're thinking on the right line, right? But I'll give you broader headings so that आप systematically अपने points इसके लिए create कर सको. One aspect is an aspect of environment, which you all are mentioning that soil can become degraded, or we don't know what kind of water requirement it will have. The ecosystem could be disturbed. इसमें एक बहुत important issue है biodiversity का, which you will read once you read environment. That if we start growing only one particular type of crop, suppose we decide कि यार ये golden rice जो है, it is a very good crop. It is a very good rice and everywhere we should use this. What will happen to the thousands of varieties of rice which are indigenously grown in India? How will the nature balance with their loss? Okay, so that can be one concern. Second is going to be a very major concern on health. BT brinjal and BT cotton, same technology, but BT brinjal did not get clearance because it's food and you're supposed to consume it. And it can have long-term health impact. So we need proper data, we need proper study, and we need to have an idea as to how this would be going, how this would be working. Alright. So because of this problem, clearance of food crops is difficult. Because you right, the impact in different ways can be different. Like if you have to eat bengan ka bharta, at what temperature will it cook, what will happen to the genes, what will happen to different material there. That is a point of concern. Then we have the economic issue. There is an issue of costs. How expensive is it going to be? Okay, how much load will this have in terms of the money component? Currently, we have a problem of monopolies. Monopolies of MNCs. Multinational companies who are making these genetically modified crops, they are monopolistic. They are the only ones who are doing business, and every time you need seeds, you have to go to them to buy the seeds. Right? So that adds an economic cost, which for a country like India is always a very, very important factor. Okay. Now on these lines, you can think of things, or इसी तरह से you can also, overall, if I ask you, for concerns or challenges to biotechnology, then again you can have all these aspects, right? There are health concerns, there are issues of cost, there are economic issues, all those things can be added. One very significant point here, which I want to specially mention separately, is the ethical issues. Can you think of the ethical issues which are involved here? What is the ethical issues? I'm not talking about crops now. I'm in general talking about the challenges to the use of biotechnology. Today, we will also be giving you an assignment question, a mains type assignment question, which will be on these lines: concerns, applications, such aspect. So, do your homework, do some research, look up into the topic and write and submit that answer. We'll give you the assignment question, so make sure you are writing that. Ethical issues अब सोचो क्या हो सकते हैं. How is ethics and morality a concern when it comes to biotechnology? Overcompensation, destroy health, patent issue, soil degradation, human health, manipulation of gene, honesty of farmers, awareness, alteration of natural occurrence. Okay, this is the first point, and a most significant point is this is a very big issue to social and religious organization. What they say is that this is you are altering or trying to alter the natural order. How nature has intended life to be, you are making changes in that. What right do you have to do that? Some religious organizations are even against manipulation of for medical use, right? Now, I told you we have concept of designer baby, right? Designer babies means I want to introduce some desirable changes into my child. So I can have a customized baby. But is it ethically correct? That's the question. Should that be done? Should technologies used be for things which are not necessary? Social and religious organization at some level, they say that not only are you interfering with natural order, but the destruction of cells also, which happens in this experimentation, that is also something which they contest. For example, when Dolly was supposed to be created, okay, or before Dolly was born, for that process to successfully go through, we had 200 plus pregnancies which failed before it could successfully be possible to have a clone being produced. Now, there is this argument that killing of those embryos is equal to murder. Yeah, that's also one extreme view. That killing of that embryo or killing of those cells, they are living specimens, that is equal to destruction of life. And nobody should have the right to do that. Okay. Then there is this issue of equity. सिर्फ ethical lines पे सोचो, बाकी तो आप challenges निकाल ही लोगे. This is an economic concept. What will equity mean? This is a game of resources, money. I have money, I have technology, right? I can use biotechnology to further become better, which will lead me to make more money. तो जिसके पास पैसे पहले ही कम थे, उसका और मेरा गैप बढ़ गया. Think of this in terms of countries. Rich countries, advanced countries, better technology, they can use biotechnology, they can become better. Like China, we just discussed, that China has the potential of, it says that it's going to make an army of soldiers with better characteristics. So, first, China already has more money. It is already ahead in terms of technology, and now with the use of biotechnology, further the gap will increase. So, ethically, it is unfair. It is a question of equity. Okay. See, for every topic which you read in all the subjects, make sure you are looking into the ethical dimension of it. Ethics जो है आपका paper 4, for that the fodder points or the content which we need, it's not going to be just from there, but they'll ask you ethics of everything. Ethics of using computers, ethics of the smartphone, ethics of international relations, ethics of shipping industry, everywhere. So everywhere, think of these aspects. All right. Okay. We'll do one last question. Then we'll conclude our session. I'll tell you all the NCERT chapters which I want you to refer. I have in between giving you other reference points also. And any questions which you have, please ask them in the doubt section. My screen gets stuck at load more, so I cannot really read everything, right? And it's a little far from me, so I have to go there and read every time. So you can post your queries in the doubt section so that before I leave, all your doubts are addressed. We have finished our biology portion. You'll have separate classes for physics, chemistry, and for the science and tech part. How can it be used for screening of new drugs? Screening of new drugs means testing of new drugs. You screen, that means you will see if the drug is okay, if it is working fine or not. So if I am making a drug for liver, I can take stem cells, change them into liver cells. I can test my medicine on that, right? And I will know if this medicine is good or not. And if it is good, I can take it to the market. If it is not doing well, if there are side effects, then we can rule that out. Weightage of bio in prelims. So Ankit, we discussed this in the first class. When you ask such questions, I feel like, the 40 minutes I wasted on the first day didn't be any fruitful. It was actually there, the number of questions was there in a PPT slide also, which you have not seen. So please make the effort of doing that. We have around 5 to 6 questions asked every year in prelims and 1 question from biotechnology in your mains exam. आंसर जो मिल रहा है वो है डी. Okay. So this is the kind of question which people struggle with in the exam. What I just want to tell you as a last footnote is that if you understand the basic concept and if you know how to solve MCQs, this question can very easily be done. But a lot of people in the real exam also got this wrong because of the stupid statements which UPSC has made. Yes, I want to say stupid because it is stupid. Okay. First statement. Genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. Right? That is correct. We can introduce changes in the eggs, in the sperm. Right? We've done a specific example of that also, which is your mitochondrial replacement, जिसमें egg ही हम change कर रहे थे. So that's very much possible. There should not be any confusion. So first statement is correct. तो अब देखो, आधा काम हो गया. Half your job is done here with this first statement. So we want 1 to be in our answer. Second statement, a person's genome can be edited before birth at the early embryonic stage. So we can do gene editing, we can do gene therapy. That can be done at the early embryonic stage. Genome is a person's complete set of genes. So you might have heard the word genome sequencing also. So if you are studying all the genes of a person, that would be genome sequencing. That means study of all the genes. If you are making an account of the whole species, so that can be human genome sequencing. कि सारे human beings जितने भी हैं human species में, all their genes you want to check. If you want to check the genes of COVID-19 virus, so that would be viral genome sequencing. ठीक है, genome means complete set of genes. A person's genome can be edited before birth in early embryonic stage. ठीक है, ये भी सही है. That means we want 2 in our answer, but there is only one option which is where it is there. So the answer is B. Very simple question if you do not read the last. Once you read the last statement, then you start second guessing. The question says, human-induced pluripotent stem cells can be injected into the embryo of a pig. Now the problem is this is not given anywhere. No news, no coverage. Probably this is something which China has done, right? No current affairs, nothing, but simple logic and option. That is what is going to help you deal with questions like this. Otherwise, ये आप सोचो कि कहीं पे पढ़ लोगे, बिल्कुल नहीं होगा. You read somewhere in detail about a pig research, next year they will ask you about the camel. Then what will you do? Then you'll again think, पता नहीं यार camel में होता है कि नहीं. So you have to be very certain of the concept. You have to be very sure of the idea and take MCQs as MCQs. I keep telling you this every time right from the first class कि question solve हो जाएगा बिना pig के भी. You don't need the pig to get to this answer. The statement is correct, right? We are giving, like I was telling you earlier, we are giving organs, we are giving cells. So this technically is possible, right? Of giving of human cells. अगर pig के human में दे रहे हैं, human के pig में भी दे सकते हैं. That's very much possible. But these are the kind of statements which derail you and you mark the wrong answer. Please don't do that. Please practice MCQs. The science part, the biology part now is very conceptual. There is no amount of theory which you can go into and you can feel ki haan, I'll handle this paper. Unless and until you understand how everything is working, it will not work out. Okay, so please make sure you have understood everything. You can go through the classes again. Please read the basic NCERTs which I asked you to read. I'll mention all the NCERT chapters. Everything from 6 to 12 today I'll summarize. Some I would have mentioned, some I might have missed, whatever it is. अभी एक बार मैं सारे बता रही हूँ, आप नोट कर लो. These are new NCERTs, the latest edition 2019 reprint. और क्वेश्चंस अगर कोई भी हैं तो प्लीज अभी डाउट सेक्शन में पूछो. The batch coordinators will pass it on to me and we can take it before we end. See, there is a prominent plant portion also. Some of it you will do in when you study geography, different climatic conditions, and some in environment, and the other basic aspect that's a little bit easier than the animal part. So you can study it. I am, however, mentioning the chapters of plants also. They are given in the 6th to 8th NCERTs. So you can do that. Actually, chapter. You have a lot of time before your 2023 prelims. So because you have started early, I hope you make the most of that time with strong bases, a good foundation. Eleventh and 12th, again, see, this is for your reference. Okay, this 12th standard, this is entirely on genetics. Okay, except for this one chapter 8 on health. All of this is on genetics, biotechnology, but it is at a very advanced level. We do not need all those details. Okay, so what you are supposed to just do is use this as a reference. जो topic आप पढ़ रहे हो, or if you come across a topic in your current affairs and you want to go and read some theory, so that's where you can read it. Okay. Other than this, just go for what we did in the class. Make sure you've covered all that topics, all those topics. You can go through these NCRTs. These chapters you will easily understand. Isme selectively you can pick out portions which you think are relevant, which you are reading in the newspaper, which you are reading in the current affairs. Then for your learning part, for your ratta part, focus on current affairs. That's going to be the zone. Usko samajhna, usko understand karna for the sense of questions, but the basis of learning, the facts, everything has to come from current affairs. Okay. I'm going to also share my email ID with you in case at any point you need to get in touch with me. jasmine.lakshya@gmail.com. You have the videos, you have the synopsis for your help. You have the talk to expert section to ask your academic queries, right? And in terms of making notes from these NCRTs, yes, if you think there is something relevant which is present and which is related to the topic which you are doing, add it to the notes which you already have in the class, right? Ye jo hain 6th, 7th, 8th, they are very simple. You might not even need to read them again. You can just summarize them and add them to already whatever place the topic is. Okay, like maybe this is about digestion, respiration, circulation. तो आपने अपनी notebook में कुछ लिखा होगा, वहीं पे add कर लो whatever additionally you find useful. Okay, so with that we are going to finish. Thank you so much for your extended patience today. I wish you all the best in this subject as you go on preparing. Special thanks to my team of batch coordinators also for the extra time they put in. If we do not have any queries or anything, we can conclude here. No, I'm not going to teach you physics, chemistry. That's way out of my league. So no. Thank you. Have a nice day all.